we talked about lipids. Okay? Lipids are a class of biological molecule defined by the fact that they have amphiphilic or amphipathic. These are interchangeable words. Okay? They mean exactly the same thing for the purpose of this class. They have dual character, so they have a hydrophobic part. Generally, we're talking about hydrocarbon rings or chains. So they're typically drawn as little tails here. And then they have some hydrophilic part. Could be charged, might just be neutral but very polar. And so we'll typically have some sort of head group. And this is our simplified schematic of a lipid molecule. Okay. Glycerophospholipids were the most common type of lipid found in biological membranes. Okay. We care about biological membranes, so we care about this type of lipid most. So here's how membranes are typically drawn for us in the text. Okay, we'll have our polar regions on either side of a bilayer membrane, and then the tails are interacting with each other in this hydrophobic region. Our lipid bilayers are about 35 angstroms, this hydrophobic region. That's the size of this here. Okay, so we have our charged groups. We have our chains in the middle. Glycerophospholipids have sort of four parts, if you will. They'll have their R group, so some charged or polar group at the top. That's connected to a phosphate. Phosphate's connected to a glycerol. Glycerol's connected to the fatty acid chains. Okay? I don't need you to know any of these head group names or memorize their structures. That's not important to me. The overall structure, this sort of four-part thing, and how they might organize themselves is what's important to me. So what was the definition of fully saturated? Fully saturated, what did that mean? Yes? Perfect, no double bonds in the chain. And that's kind of illustrated here. It's just a straight zigzag. If there were double bonds, we would see a second line illustrated somewhere. Could the chains have double bonds? Yeah, absolutely. Okay, could be unsaturated. Which would be more reactive, saturated or unsaturated? Okay. Lauren? Why? Perfect. Great. So unsaturated would be more reactive. This is a site of bonding or something, a site of attack that something might want to come in and interact at that double bond, take it down to a single bond. Which would be stiffer, saturated or unsaturated? Why? Which bonds are easier to rotate, single or double? Single. So if I have all single bonds, there's maybe a little bit more flexibility in those tails. As soon as I put in a double bond or several double bonds, I probably can't move those tails quite as much anymore. That rotation goes away. So these might be a little stiffer when I'm trying to make lipid structures out of them. Okay, they might not be able to pack as neatly or as densely. So both of these things would influence the physical properties of whatever lipid structure I end up with, whether it's a bilayer or, and we'll look on the next slides, the other ones we talked about, micelles or liposomes or vesicles, whether the chain has double bonds in it or not, how long that chain is, how many chains there are. All of these things will influence the physical properties of that lipid structure. Charge on the head group, whether it's actually charged or it's just neutral polar would influence it. So all of these things. I got a good question last time that's a bit outside the scope of this class. Where's my mouse? I apologize for just doing this a whole bunch. I never know where it is. I got a good question about whether or not you could model sort of a bilayer or any of these lipid structures sort of using beam theory or as like a mechanical beam, which is something that gets covered in 418. And the answer is 100% yes. And I can put some nice resources about doing that. So if you're not in 418 or you haven't really heard of that, don't worry about it, but you definitely could model sort of the structural surface of one of these lipid structures like a beam and look at its mechanics that way. It gets really complicated when we think about the true bilayer, though, because it has so much stuff embedded in it that's changing its properties. It's not like a smooth, even surface of these molecules. Okay. So the key behavior of lipids is that they have a strong tendency to phase separate spontaneously when they're in water. So and the way to think of this is like salad dressing or oil droplets in water. Okay. We have a bunch of, we have our polar head groups, single chains here, and at some critical concentration, there's some low concentration where they can stay in solution. And once you surpass that, they're going to spontaneously form themselves into micelles. When you have double chain lipids, this is when you're generally going to get structures more like bilayers or vesicles or liposomes. Okay? Liposome and vesicle I'm using interchangeably. And the key difference here is size. So if we think about sort of the structural dimension of the different lipid structures that I've talked about, so first of all, we have a logarithmic scale here from 0.1 nanometers, which is an angstrom, 1 angstrom, up through 1 millimeter. Okay? And so we're looking at atomic level through sort of embryonic size, and then out here at 10 or 100 times more, you would get like an actual egg, chicken egg or something. Okay? So look at lipids here. We're on the order of about 1 nanometer, maybe a little bit bigger. Okay? Proteins are a little bit bigger than that generally. Micelles. Okay? Micelles are kind of the smallest. These are roughly in the 3 to 50 nanometer range. Liposomes or vesicles, again, used interchangeably, get a little bit bigger. Okay? So now we're talking about 20 nanometers up to 2,000, or roughly 2 microns-ish is kind of the size. And of course, the unilamellar or single kind of layer ones are down at the smaller end. And when you have those multi-layered vesicles with kind of several rings concentrically, now we're up at the big size. Makes more sense. Okay? And then bilayers, phospholipid bilayers, these are like giant liposomes, or giant vesicles, if you will. They don't cut off. They go all the way around. And they're encapsulating either something like organelles, so they're all the way around the mitochondria or the nucleus, or a whole cell. So there's a wide range here, too. You know, we might be talking about on the order of 1 micron, or if we're going around entire animal or plant cells, we're up here, 10, 20, 30 microns. Okay? So understanding sort of relative size difference. I don't need you to memorize the exact numbers, but this is kind of one of the key differences between these structures. And of course, you're not going to be able to make something big if you don't have enough lipids in solution for it to happen. So concentration will dictate what you can get. Okay. So again, this is a repeat of the sentence from before. The lipids have a tendency to spontaneously form these structures on their own. Okay? Because they have those really nonpolar regions. We got to get those nonpolar regions together. We can't have a high concentration of this stuff in an aqueous environment. Okay, so shape kind of dictates what we'll get. When you have sort of one tail, it has more of this conical shape, and so that's why we might form a micelle. Whereas when you have two tails, the overall sort of shape of the individual lipid molecule is cylindrical, and so this is how we can get layers, the bilayers or the liposomes. Okay, and so when we talk about spontaneous, anything that's happening spontaneously is because it's energetically favorable. Okay? We want to lower energy. We want a net negative change in energy. All of those terms are equivalent for spontaneous. Net negative change in energy. Energetically favorable means lowest possible energy we can get to. And so, it would be unfavorable to have all of this exposed sort of edge to our bilayer. So, of course, it's going to fold in on itself and become a sealed compartment where you can have an aqueous environment inside. These figures are super blurry and bad, but if you want quality versions of them and more explanation of this slide, this is a good link for you here. Okay. So these were questions from last time. So one of the questions was, how might curvature affect membrane structure? Okay, so which structures out of our choices here, what was the least stable? Micelles, why? There's a lot of reasons why, but let's talk about curvature exclusively. Sure.
It's so curved that when collisions happen, it's easy to break it up.
Okay. More exposure to sort of like thermal energy and just collisions could break this apart. That's a really good point. It kind of goes with the fact that you can kind of see it here. The head groups of the micelle are literally physically further apart. The interactions between them have to happen over a wider range. And so it would be easier to break them apart. Okay? So micelles and vesicles tend to have highly curved surfaces. Bilayer membranes do not. In fact, if you wanted to model a bilayer and you want to go really simple with it, they'll often model it just as flat. It's obviously not flat, but we go ahead and say, okay, at the scale we're looking at, it could be assumed flat if we took a very small micron scale region out of it. And so, true or false, covalent bonds form between the adjacent head groups in the bilayers. False, great. Okay, so the polar head groups are making non-covalent interactions, some sort of electrostatic interaction, maybe it's a true hydrogen bond, any of those non-covalent options. They're also going to be interacting with the water and the polar environment around it. Okay? And so the reason we don't have a true covalent bond, these lipid molecules are moving dynamically. They're free to diffuse laterally in the membrane. They have a couple other sort of movement options. So they can move laterally. They can sort of flex in their tails. They could rotate. They can flip-flop sides. And you could track all of this. I won't ask questions about any of the details of sort of fluorescent imaging or tracking the movement of lipids in the bilayer. Okay. But be aware that they can move and that they are not making covalent bonds. Okay, so we also looked at this figure last time. We sort of said the bilayer is really oversimplified when we think about it as just lipids. The reality is, of course, we have proteins embedded. We have other types of steroid or lipid-style molecules, cholesterols. We have proteins on the inside anchoring the membrane in place, so it doesn't have quite the level of flexibility that we're imagining a micelle or a liposome would have. Okay? And the composition, sort of its thickness, its surface charge, what other molecules are embedded, control physical properties. So biological membranes, lipid bilayer, kind of the structure we'll talk about most often. Not that micelles or liposomes wouldn't be cool for things like drug delivery or time-release delivery or some transport. But when we think of membranes, that's what we'll think about most with our lipid structures. They're designed to be barriers. The idea is don't let things through. Separate ions, protons, signaling molecules, keep things separated in the different compartments of the cell because they have different functions. They need different concentrations of these molecules and ions. Okay? And so we have membrane going all the way around the cell and then, of course, separating out different key compartments. And this is to keep sort of polar things across. It would be a really high energetic penalty to push something charged into the hydrophobic tails all the way across. So meant to be a barrier. In kind of a bizarre way, though, what biology does is, 1, set up all the barriers, 2, immediately find a way to get across the barriers. So that's kind of what we end up having to do, and that's the function of these membrane proteins, right? So we set up this great hydrophobic barrier to keeping everything out of different compartments, but we know that functions of our cells and of our organelles require transport and signaling across all of these membranes, so we'll go ahead and embed proteins in them in order to accomplish this goal. And so our membrane proteins sort of have generally 2 key domains, the part that's actually traversing the membrane, and that part needs to be hydrophobic to interact with the tails, and then usually some peripheral region. And the peripheral regions could be on the inside of the cell, the outside of the cell, both sides of the cell. Can look a couple different ways. And then those are going to be hydrophilic because they're back in an aqueous environment.